Okay, thank you very much for the introduction, Sam. And so I'm very lucky that yesterday many people gave the background information, which I don't have to repeat, so I can go very fast over the first few slides, but I'll show them anyway. So you already saw a few times this famous cartoon of how the cells move on surfaces. There are basically three processes. There has to be a protrusion at the leading edge of the cell. There has to be graded adhesion at the interface between the cell and the surface it moves on, so that adhesions are firm at the front and relatively weak at the rear. And then there has to be contraction at the cell body, which pulls forward the cell body after the cell makes an extension. And as people yesterday told you, there are many instances where cell motility is absolutely essential in cell biology and physiology, so I'm not going over it too much. Hans already showed you this famous movie of neutrophil chasing the bacterium, right? It's all over the internet. On YouTube, you can see lots of comments on this movie, from which you can easily glimpse how much the public appreciates science and understands science. There are many more there. Anyway, so what I will tell you three stories today. The first is going to be longer, the second very short, and the third relatively short. And the first two stories relate to fish epithelial keratocyte, which is our model system of choice. People worked on it for decades. As was already explained yesterday, there are few advantages to it. So one is that keratocytes, when they are alone, when they're placed on a flat surface, they move rapidly with a very steady shape, with very steady velocity. What you just saw was the cell moving pretty much straight. In the front of it is the lamellipodium, which was discussed much yesterday. This is a relatively flat network of actin. And at the rear, the cell body, you can appreciate the geometry if you look at the side view of the keratocyte. So the lamellipodium is incredibly flat, and cell body is this three-dimensional mound. For understanding mechanics, cell body is pretty much irrelevant, because as experiments showed, one can generate lamellipodial fragments which move and behave pretty much like the mother cell. So there's really nothing in the cell body which is part of the cell engine. So one thing I wanted to say in the very beginning, I will come to that also in the end, that there's no the mechanism of cell motility. There are tens of different mechanisms which different model organisms combine in different circumstances differently. So what I will be telling you today is not the absolute truth. This relates only to the keratocyte, and it's an open question how it is useful for cells moving in 3D, for other kinds of cells. But I'll come back to this point later. So this people already showed you yesterday also. So molecular machinery of the lamellipodium. There's this famous treadmilling model for actin filaments growing at the leading edge, pushing the leading edge forward. These filaments age, and aged parts crumble down, disassemble, then monomers, actin monomers reassemble onto the leading edge. And all of this is mediated by a vast host of actin binding proteins, including ARP23, which people already discussed, capping protein, which stops growing from growing too long. So this is a completely separate topic which can take hours, and I'm not going to talk about protrusion in detail today at all. What I will focus on is the very important part of motility which was not discussed much yesterday, and this is contraction. And contraction is mediated by myosin. So there's actin-myosin contraction at the rear of the lamellipodium. There's another discussion which could run for hours, the difference between the lamellipodium proper and what's at the very rear, which more properly should be called lamellum. But for largely mathematical audience here, this subtle difference is not super important. But basically, what Gary Borisi and other people proposed almost 20 years ago, the qualitative picture of how lamellipodium operates, turned out to be pretty much true. And what we spent the last 20 years on is trying to substantiate this hypothesis and make it more quantitative. And the idea is basically as follows. At the leading edge of the lamellipodium, actin network assembles. And this network is very dense, it's highly cross-linked and branched, and mechanically it's relatively firm and stiff. And also there's a very firm adhesion at the front to the substratum. And then elsewhere in the lamellipodium, assembly still goes on, but disassembly prevails. And so there's net disassembly of actin network across the lamellipodium. And so what happens is by the time any specific material point of the lamellipodium reaches the rear, the network becoming sparse and mechanically weak. And so there's also, as I mentioned, myosin in the lamellipodium. And myosin starts contracting, crumbling, and breaking the remaining weak network of the lamellipodium. And so at the rear, the contraction completely dominates. And so the idea is that protrusion is at the front goes forward, contraction is at the rear, and this contraction pulls the rear forward. And this very same contraction somehow contains the sides of the lamellipodium from running away. So that's the qualitative picture of what's going on. So the interesting thing about this actin-myosin contraction is that it's from the kind of bird view, the same process that happens in our muscles. But in muscles, there are perfect crystalline actin-myosin arrays, while in the lamellipodium, the contractile actin-myosin structures self-assemble. And how it's happening microscopically is still very much an open question. And microscopic nature of contraction I'm not going to touch. What I'm going to discuss is how this contraction organizes the cell.
I don't know whether if it's too long. So the cartoon you are suggesting is starting from the center. Mm-hmm. There's like an extensile dipole in front and a contractile dipole in the bottom. Is it?
Yeah, that's fair. Yeah.
So two dipoles instead of one.
Fair description. Yeah. Okay, so what I'm going to focus on today, is the phenomenon which has fundamental biological significance and also is irresistible for physicists and mathematicians, which is polarization and initiation of the spontaneous motility of the cells. So what you see in this movie is initially when a keratocyte cell is placed on a stiff flat surface, the cell looks like a fried egg from the top. So lamellipodium makes this flat ring around the periphery, and at the center is this mound of the cell body. And then spontaneously, so people did this experiment very carefully many times, there are absolutely no directional cues from the environment. Spontaneous, the cell polarizes, assumes this characteristic fan-like shape and starts moving. And so the question is, what is the nature of this symmetry break? What is the nature of this phase transition, however you want to call this phenomenon? So what I'll tell you in the next few slides is how we understood this phenomenon using the combination of microscopy, pharmacological inhibitions, and mathematical modeling. And so let me introduce you the simplest, perhaps the simplest possible model of the mechanics of actin-myosin contractile lamellipodium. So mechanically, we model the actin gel as a viscous fluid, highly viscous fluid. So it could be endless discussion about the actual mechanics of the actin network. Some people prefer to model it as elastic material. some as viscoelastic material, viscous. Viscous, it's a big and complicated subject. Basically, the simplest thing I can say, experiments show pretty much unequivocally that if you deal with time scales longer than a few seconds, and in this case, the relevant time scales are tens of seconds and minutes, then actin network is pretty much viscous. The reason for it is that it's cross-linked by, as I said, many actin-binding proteins, and these proteins bind, unbind on the scale of 1 second. Okay. So if you start deforming this network on the scale longer than a few seconds, every individual actin filament starts unbinding and flowing relative to other actin filaments, and the network becomes pretty much viscous. It's not necessarily Newtonian viscous fluid, and it's certainly compressible. It's not incompressible. What we know about keratocyte is that we tried to model it with viscoelastic models before, like with Oldroyd B models, and we found, comparing with experiment, that elastic parts is pretty much irrelevant. So another huge topic is what is the pressure in the lamellipodium, and whether it's compressible or incompressible, and it's also complicated and open subject, and that I can discuss with you in the discussion. It's too long of a subject. Basically, for now, we have just viscous stress in actin network. So here, vector U is local rate of actin flow. This is how fast material point of actin network flows relatively to the surface.
And it's time independent?
It's both time and coordinate dependent. Yeah. Yeah.
And does the cytoplasm play a role here?
No, in this model there's no cytoplasm. So that kind of goes back to this pressure and all other things, and that I can discuss later. It's an important point.
And how about the membrane?
No membrane in this model. So all that we use, but I'm not going to show this in the equations, is that it seems that in keratocyte cells, membranes stretch to the limit and there's no intracellular membrane anymore. So largely the total area of the cell is conserved. And this is kind of hidden in our assumptions about protrusion. Yeah.
In your picture above, you've got two regions.
Two regions?
Yeah,
The
you've got your
nucleus
ellipse.
and the...
Oh yeah. So we don't model, well, we model the nucleus a little bit, but not.
This is just ignored.
It's largely ignored. Yeah.
So these equations are holding in that.
In the flat.
Just in that two-dimensional.
Lamellipodium, correct. Mm-hmm.
And that's moving.
Right. Mm-hmm. All right. So there are two equations in the model. The first equation is the force balance, and the second equation is the transport. So the force balance. There are balance of three forces. There are many others, but those three forces are dominating. Passive viscous stresses in actin network, then myosin contractile stress. In fact, right. So the idea is that myosin, we assume that myosin stress is simply proportional to the myosin density, which is an assumption, and it's probably not exactly right, but in fact, myosin stress is, of course, growing function of myosin density.
So what do you define as stress? Is it a force, or what is stress?
It's a stress. It's mechanical stress. It's a force divided by a unit surface across which this force is applied. So this is two-dimensional model. So then it's force divided by the unit length.
It's isotropic.
Right. So the assumption is that myosin stress is isotropic, right? Which is also a big assumption. It's not exactly true. It should depend on anisotropy, local anisotropy of actin network. In this model, this is completely ignored. There are many indications that so when you start comparing with experiment, that anisotropy doesn't give you much interesting information. So then gradient of this myosin density is local force per unit length. There's some proportionality coefficient which basically shows you how strong one unit of myosin is. And the third force is adhesion viscous drag. So actin network flows relative to the surface there are dynamic adhesions between actin network and the surface. And the mechanical nature of those adhesions are not well known. So the vast majority of models so far assumed that this friction can be described by the viscous drag. So force or stress is proportional to the local velocity, and zeta is the adhesion strength, the proportionality coefficient. So this is the force balance. Then we of course have to know what's the myosin density, and myosin density is dynamic and is described. Yeah.
So you don't have an equation for actin density?
No. So yeah. So in more complete models, we have additional equations for actin density. I'm showing you kind of the bare minimum which can explain the phenomenon. So myosin density is dynamic, and in the simplest model, it's a simple diffusion drift equation. Diffusion part is not important. We keep diffusion very small, mathematically just to smoothen everything, but biologically there is some diffusion. So largely speaking, myosin simply moves with actin. Myosin clusters bind to actin network and flow with it. But then, of course, myosin molecules unbind, bind to some other place, and this unbinding, diffusion in the cytoplasm and binding to some other place, you can do some perturbation theory and show that effectively this is diffusion.
Don't they also walk on actin towards plus n?
Right, they do. And so, because the model is completely isotropic, this is effectively goes into diffusion. So there's lots of complications you can kind of pile up on top of this model. Okay, so those are two equations. So what I want to emphasize about them qualitatively is one is what is in the right-hand side of this equation, the adhesion viscous drag, is what is called traction stress. This is the force per unit area applied by the cell to the surface on which they move. And Eva and some other people yesterday discussed it a lot. So this is the traction that can be measured if you put cell on deformable surface. Another thing is what you can easily glimpse from this system is that there's very important positive feedback here between myosin and actin flow, right? And qualitatively, it's working in a very simple way. Imagine that you have aggregate of myosin at the center of the cell. Then myosin will create this net forces such that the force is trying to move actin network into the aggregate of myosin. So myosin is moving actin network up the gradient of myosin density. And then this flow of actin brings even more myosin to this aggregate, which create even higher forces and higher flow, and on and on and on. So for mathematicians in this audience, there are many analogies of this model to the fundamental Keller-Segal model for chemotaxis. Right? So myosin effectively is like bacteria secreting the some kind of chemical, and then other bacteria move up the gradient of this chemical. So Hans did some pioneering work decades ago on this model, and so there are many parallels here with that model.
I'm sorry to pile up another complication. If I were to measure myosin flow, let's say by bleaching and observing over minutes, I have an average diffusion and drift.
Right.
But I fear I wouldn't measure what you model here because we also have activation of myosin somehow folded in.
Yeah.
How do I compare that to an experiment?
Yeah. So what we did, I'll show you some experimental data. People looked at myosin light chain, phosphomyosin. For this crude level of description, it pretty much doesn't matter. But there are indeed more complex models when you have to model how individual myosins get into clusters, how they phosphorylate with all this stuff. It's yeah. But so if you do photo bleaching, you will observe what this model predicts. Those experiments were done.
What are the boundary conditions?
I'll show that. Great question. So the first thing we did, we took this model and made two simulations. So You saw already in this polarization movie that the cell first is like a disk. Yeah.
In the model, where's the direction? When the model, the direction of the myosin is generated. You say that
There's no direction. There's an isotropic contraction.
but in what direction? Okay, you say you do not have a lamellipodium at the moment.
Sorry?
You do not have a lamellipodium
Right.
kind of environment at the moment. You just have an isotropic room
Right.
where your model is
Right.
running in.
Yeah.
Okay.
No
Mix
direction.
of initial conditions or boundary conditions determines.
I'll show you the rest. So we started with doing these two simulations. You saw in the polarization movie that the cell initially is like a disk, and in the end it has this fan-like shape and it's moving. And so the question is, what would this model predict in those two simple geometries? So in the disk-like geometry, the predictable thing happens. Myosin starts to accumulate at the center, and it creates actin flow, which is centripetal and radially symmetric. And this flow brings even more myosin to the front. So one complication that we did, but try not to think too much about it, it's not important, is that we did have cell body in the simulation, and cell body is simply kind of repulsive potential at the center of the cell. But that doesn't really matter. But that's why you have this light shade here. So we did it to compare results better with the experiment in which the cell does have the cell body. So it's very simple result for the cell which is symmetric and sitting on the surface, and for asymmetric cell which is moving. Right? The key thing is that myosin is stockpiled at the rear, and that's very easy to understand. If you are sitting in the if you observer sitting on the moving cell, what you see is how actin network goes away from the front to the rear. And this flow blows all the myosin to the rear. That's why it's stockpiled there. This is again the shade from the cell body. And that's where myosin create the most of the flow. And here's the predicted flow. It should be strong at the rear where myosin density is high, and also the gradient of myosin density is high. And so both of the experiments, numerical experiments, were done with completely uniform constant adhesion strength. So, yeah?
You just change the geometry of your domain between the two
Uh-huh. Yeah.
and that induces a difference.
So geometry plus, in this case, the cell is moving with constant rate this way, but basically in the simulation there was additional flow of a kinematic flow of actin to the rear. Right? That's it. That's it. So these simulations reproduce pretty well the qualitative observations for the flow and myosin. So in the stationary cell, actin flow, which was measured with fluorescent speckle microscopy, is indeed centripetal, going from the periphery to the center. It's of course not radially symmetric because nothing is uniform and nothing is constant in the live cell. In the moving cell, as predicted, actin flow is fast at the rear and slow at the front and the sides, and also centripetal. Myosin in the stationary cell is aggregating not of course to the center because the cell body is sitting here, but to the periphery of the cell body. In the moving cell, myosin is blown to the rear of the cell.
Sorry, in the actin flow in the moving cell, is that in the lab reference frame or in the cell-based reference?
In the lab frame. So that's a great
And
question.
how long is the velocity vector of the cell compared to the actin flow at the end?
Also, excellent question. It's the speed of the cell is of the same order of magnitude as this rate at the very end.
Thank you.
And where is the disassembly of actin occurs here? Actually,
So in the model we have no actin, but in the more complex model there is actin, and so it's known experimentally, and also the models show that actin disassembly is everywhere, but it's strongest at the very rear. So it's a famous experimental result from Thiriet lab that disassembly pretty much proportional to local myosin. Yeah.
The check in your simulation keeps the cell wide. It also could be possible with some manipulating your simulation that the cell would be a long cell with a small lamellipodium and a long front.
Right. So then qualitatively things would be relatively similar, but it's a great question. But anyway, I will show soon the free boundary simulations. So. Okay, so these simulations on the fixed shape, only part of the picture, of course. What would be really interesting is to see if this model, what this model shows if we free the boundary, right? Because the idea would be that the shape of the cell has to evolve dynamically. It's not imposed by the cell by some in some way, but it's mechanically evolves.
So those pictures you showed, you're imposing a motion on the domain.
Right. So those simulations on fixed shapes, it's just solving PDEs on a fixed boundary. So, but now what we want is to do it with a free boundary. Okay. So what we have to do before we solve this free boundary problem is to introduce boundary conditions which were also actually the same in fixed shape and rules for movement of the boundary. So the boundary conditions are for Myosin flow. It's simply no flux of myosin through the boundary, right? Very natural boundary condition. And for the force balance equation, it's zero stress at the boundary, which is also a highly debatable condition. And in fact, we are now doing a couple of projects showing that the solutions are highly sensitive to the boundary conditions, and that I'm not prepared yet to discuss. But so this is simply done because this is what people so far used a lot.
So this is if there wouldn't be no membrane.
Well, so that's exactly the point. The thing is that there are membrane tension everywhere, right? And membrane tension should impose some constant stress. But whether this stress should be fed directly into macroscopic equations, that's a big question. So there is a stress on these individual filaments pushing the membrane, but whether so it's largely applied to the adhesions which are at the boundary, and yeah. So what I can say, if we have some constant here instead of zero, nothing pretty much is going to change. But if you start playing with something more complicated, then things could get very complicated.
Is the continuity equation is
Continuity
at the boundary? What's the velocity of the
Yeah. So right. So this is for moving boundary.
So this is okay.
So the rules for the movement of the boundary are very simple. First of all, the boundary simply moves with the local actin flow velocity imposed by solving this two-dimensional system of PDEs. So basically, if there was no actin protrusion, if there was no growth of actin at the boundary, the cell would simply start collapsing and collapse with the rate predicted by myosin contraction. As simple as that. So in the locally normal direction, then the boundary moves with the projection of the predicted rate of flow onto the locally normal direction. But of course, what's happening at the boundary, what people discussed a lot yesterday, was that actin network grows. And so in this model, the assumption is super simple, that the growth of the boundary is a constant, happening with constant rate in a locally normal direction. Okay. So where's this assumption coming from? There was famous experimental paper more than 20 years ago showing that at the leading edge and at the sides, the growth of actin network is happening in the locally normal direction. And there's some fundamental physics behind it, which will take too much to discuss. The fact that this rate is constant, independent of the position on the boundary. So this is known for symmetric cell. When the cell becomes asymmetric, the growth rate of actin network, of course, stops being constant. And this is probably an important part of polarization of the cell. But in this minimal model, for simplicity, we keep actin rate constant just to show that the key mechanism has nothing to do with actin growth. It has to do with actomyosin contraction. And so each point of the boundary simply moves as a geometric sum of those two rates, protrusion and contraction. And so then numerically...
is U perp the normal component of your velocity field in the domain? Is it?
So, sorry?
Is U perp, your local, is the normal component
correct.
of the...
So we solve this problem numerically using the level set method. It's numerically relatively challenging problem, but it's not, it's almost routine these days. It's not, it's not
So
too
can I ask
hard.
another question? Your parameter nu, the local protrusion rate, is that just a number?
Oh, no. So VP is the number. This is local protrusion rate.
Well, I'm
and
sorry.
It's constant. And
What's the normal?
just the sum of VP minus U normal.
I see. I don't really see.
Okay, so let me first show you the snapshots from the simulation with free boundary. I will later show you the movie. So we solved this model on a free boundary domain. And so the simulations were set like that. Initially, we created gradient of myosin. We shifted most of the myosin to one side of the cell, which becomes the future rear. And just saw what happens in the free boundary simulation. What happens is that you see that the cell shape remains pretty much symmetric. There's a super slight asymmetry. It's actually ellipsoid with epsilon less than 1%. And the cell creeping forward, it's moving forward really slow. So you see that there's still a really slight gradient of myosin at the rear, and there's slightly higher inward actin flow at the rear. So what happens is very simple. The cell starts moving, which sweeps the myosin to the rear, and then movement of the cell keeps myosin at the rear. But why is the cell keeping moving? The reason is that higher myosin at the rear creates higher flow, and effectively the net rate of the boundary movement is this way in the rear and this way at the front. So just this one positive feedback between myosin and actin flow can actually generate very slow movement of the cell. So in principle, the polarization can be explained by just one positive feedback. But in fact, of course, it doesn't show what the experiments show. We don't have the drastic change of the cell shape, and we don't have the observed velocity. Okay. And so we're missing something. What we're missing is hinted at if you start comparing the traction forces and the actin flow in the stationary and moving cell. So those are the experimental results. In the stationary cell, what you see is that the flow is slow and the traction force is high. And in the moving cell, what you see that in general, the flow is higher than in stationary cell, right? There's this high flow at the rear. But traction is pretty much low other than in two hot spots at the rear corners of the cell. Okay. I will get to these regional differences later, but so far the global picture is that the traction is higher in stationary cell and lower in moving cell, and flow is higher in moving cell and lower in stationary cell. So what the model is missing is pretty obvious. Traction in the model is adhesion strength times the flow. So adhesion strength is not constant, which of course many people in the audience could guess from the beginning. So we should change this assumption and see what happens. So what should we assume about the adhesion strength? So the hint came from the fundamental work from Gardel and Waterman-Storer about 8 years ago, where in the different cell in the fibroblast, they observed the phenomenon which is now being observed in many other circumstances. That if you start simultaneously measure traction stress and rate of actin flow in different points, you will see in some regions of the cell the region where traction is proportional to the actin flow, so it's pretty much this viscous drag, right? But then at higher actin flow, the traction suddenly starts dropping, so there's this biphasic dependence between the flow and the traction stress. So the simplest interpretation, and it's not yet clear that this simplest interpretation is right, that adhesion has this stick-slip property. And different people put different meaning into this stick-slip idea. But so this is kind of naive, simplest stick-slip of the following kind. Imagine that adhesions are sticky springs which start detaching at some threshold rate of movement. So if actin moves slowly relative to the surface, most of the springs are attached, and there's a high traction, high adhesion strength. But when the flow becomes fast, springs detach, and there's no adhesion. So you pretty much rip off your adhesions. So that's kind of the simplest interpretation. As I said, there's actually much modeling experiment now on the adhesions. People try to understand if this is true or not. So that's what we started to assume. So the assumption now in the model is that adhesion strength is a function of the absolute value of the actin flow, and it's this simplest possible dependence. So there's a high adhesion below the threshold flow and low adhesion above the flow. So when we computed the traction stress with this assumption and plotted it as a function of the actin flow rate, and then we measured in the keratocyte the traction and the flow and found the ratio of the traction to the flow, this is the experimental data which is kind of reasonable with the theory. Okay? Yeah.
Do you measure the traction stresses point-wise around the
Point-wise. Right. Okay. So qualitatively, what we just did, we introduced the second feedback, which is negative. We had the positive feedback between the flow and the myosin. Now we introduce the negative feedback between the flow and adhesion. So it's as simple as faster flow weakens adhesion, stronger adhesion, lower the flow. So we have now a combination of positive and negative feedback. And so together with those two assumptions, this is the free boundary simulations. So we start with symmetric cell, and then it evolves into this characteristic shape with the correct distribution of the traction forces, and the cell is moving. So in this simulation, we also had some noise. So this is stochastic simulation. Otherwise, you cannot start. Well, you can get this shape from asymmetric initial conditions. So I'll show you. So what the stochastic term was, we added white noise to the actin flow. And it's a long discussion why this is the best thing we can do now.
But is it through the initial conditions?
So this is for uniform additional conditions, uniform isotropic, with noise.
That's space-time white noise?
Sorry?
Is that space-time white noise?
Yeah, so it's space-time white noise, yeah.
Alex, when you are speaking about adhesion, it is continuously distributed over the whole surface?
Correct.
So not like in reality?
No, not like in reality, not the discrete adhesions.
I can't quite tell from back here in the colors. Can you verbally describe your velocity direction vectors? I can see the reds in the back.
So there's pretty much centripetal flow inward. There's of course the flow is not locally normal to the boundary, but roughly speaking it's centripetal. So this is traction. I have separate movies for flow, for everything. So the qualitative explanation for this phenomenon is very simple. The initial fluctuation creates higher myosin at one side of the cell, or adhesion is lower at one side of the cell. Then at this side, the flow becomes faster. The flow rips off adhesions there, and the flow becomes permanently high there. And so this side becomes the rear. That's it. Then the cell moves, keeping myosin blown to the side, kind of locking the cell in this state. So we tested this model.
The cell is not allowed to move. You just keep it fixed and round. Does the combination of myosin and actin on adhesion, all that, produce a reorganization of actin, or it stays more or less the way it was initially?
This is a great question. I was actually going to show it at the end in the different project, but then if the cell is fixed, you cannot polarize it. So the movement is essential part of this process.
Does the actin reorganize at all, or hardly?
Well, actin, not really. It remains largely symmetric. So there are many tests we did. Let me show you just a couple. So this is the example of simulation when we started with. So this is without noise, and this is with asymmetric initial conditions. We lowered the adhesion at one side of the cell initially, and then this state evolves where the cell keeps this fan-like shape, moves this way, adhesion remains strong at the front and the sides, remains small at the rear, actin is blown to the rear, flow is high at the rear, traction is high at the rear corners as an experiment. And this is the experiment in which RGD solution lowering the adhesion is blown to the cell locally to the one side of it, and this side becomes the rear, and the cell starts moving away from the pipette. Another test, you can do the same thing initially, shifting myosin a little bit to the rear. The very same final state evolves, and the experiment is you start blowing colliculin, strengthening myosin at one side of the cells. The cell recoils from the pipette and starts moving. So that's pretty much the first story, right? And so for biologists, the main conclusion here is that in keratocytes there are two essential feedbacks: one positive and one negative. Negative feedback is stick-slip adhesion, and positive is actin flow myosin, and so together they give you the polarization.
So mathematically, could you identify a bifurcation parameter that could transit from stationary?
Yeah, that's a great question. We didn't do that, and this would be a very worthy problem. I would do it in 1D model. So 1D model is almost analytically solvable for this equation, and so there you can work with this bifurcation. So Pierre Recher here actually did some very cool work on that. Yeah. Pierre, you had a question.
No, I just...
Okay.
Yeah, we can discuss about that.
So this lamellipodium is presumably is dragging the nucleus with it.
Right.
Which must be significant.
Yeah, it's a great question. So all indications are that so for nucleus, all of this is very important for nuclear position. For the lamellipodium, where nucleus is probably not important in the keratocyte, so we didn't do anything about the nucleus. In 3D, it's becoming super important, but yeah, this is 2D problem.
So have you looked at 3D?
No. We didn't. So in the last project, I'll mention we did experiment in 3D, but we didn't model it in 3D. Okay, so in the last 10 minutes, let me just glide over a few slides. So in those two last projects, probably hold on your questions until the discussions. As usual, the first story took much longer than I thought. So this was spontaneous motility initiation. Now, what happens if polarization is induced? So this brings us to the fascinating subject of how cells behave in electric fields. So keratocytes are not known to be chemotactic. It's not clear if they are not chemotactic at all, or nobody found chemical yet that attracts them. But they do move along the electric field. And so in our lab, we did this experiment, which actually was done many times before. So keratocytes on the cover slip move to the cathode. Then you switch the direction of electric field, which will happen in a second. click, then they repolarize and move to the cathode again. And this is repeated. All cells do that. So all cells move along the electric field, some to the cathode, some to the anode. And there's a vast literature behind it, which barely attracted yet the attention of cell biologists and mathematicians. Yet it's a fundamental part of physiology. For example, in wound healing, in wounds, there are strong electric fields, and I don't have time to discuss how they arise. And so the cells move along the electric field into the wound, closing the wound, and this is a very important process. There are many experiments show that if you disrupt electric field in the wound, it doesn't heal, well, it heals order of magnitude slower. And barely a research topic, which I would say will be a major theme in cell biology in the next decade, is that electric field is a huge part of self-organization of the embryo. So there are experiments showing that if you disrupt electric field in growing embryo, it messes up cell motility, division, and so on. So I don't have time to show you our research on signal transduction related to this, how cells actually sense electric field. I just wanted to show you just the tip of the iceberg, which relates to the polarization of keratocyte in electric field. So one simple result that if you start with the same condition as I described in the first part of the talk, and see what happens in electric field, then the cell in electric field polarizes order of magnitude faster. So spontaneous polarization takes tens of minutes. Polarization in electric field takes a few minutes. But so what is fascinating is that the cell which was already polarized before the electric field applies, it remains polarized after the electric field is switched off. It just starts moving in random direction. But the cell which got polarized by electric field and starts moving to the cathode, it depolarizes as soon as the electric field is switched off. See, field is off, the cell depolarizes. So it suggests that their mechanism of polarization in the electric field is somewhat different. And the clue of what is different comes if you start looking at the distribution of actin, myosin, and vinculin, which is one of the adhesion proteins. So actin is distributed in stationary cells and in motile cells the same in the motile state and in the stationary state. Myosin is distributed similarly in motile state and in the stationary state. But adhesion proteins, vinculin among them, is distributed the same in stationary state and differently in the motile state. In the motile state, all the vinculin is blown to the rear of the motile keratocyte. But in the motile keratocyte in electric field, the vinculin creates this bright rim at the leading edge of the cell. So we don't know yet what it means, but here's the speculation. Vinculin is a marker for mature force-sensing adhesions. And so in the stationary cell, remember that there's high traction at the periphery of the stationary cell. And so there's strong adhesions there sensing this tension. And those adhesions disappear in the motile cell, which is completely in agreement with the model I showed. But so in the cell in the electric field probably kicks adhesions in completely different state, where when they remain strong everywhere, like in stationary cell, somehow electric field manages to weaken the adhesions transiently at the rear, and then the cell is able to move. Okay. And then electric field is switched off, adhesions recover everywhere around the boundary, and the cell stops. So that's kind of vague speculation. Let me leave it on that. We have actually a simulation of this, but I don't have time to describe it in detail. So this is before electric field switched on, then when it's off, the cell stops. I just wanted very briefly to show you in 5 minutes.
Just a very quick.
Sure.
If you leave the electric field long enough, does it polarize permanently?
We tried to leave it for 10 minutes, 30 minutes, hour, 2 hours. Every time you switch it off, the cell depolarizes. Then we have lots of interesting conditions when we inhibited PA3 kinase, myosin, then things change. So there's a lot to this story which I don't have time to describe. So I wanted to very briefly just run over a completely different mechanism of cell polarization, just to illustrate the point which I started with, that there's no the mechanism of motility, polarization, anything else with the cell. Different cells do it differently. We just finished the project with different type of cells, with red liver cells, which when they are stationary on the surface, they look pretty much like that. Very similar to the keratocytes. There's a nucleus at the center, but the huge difference from the keratocytes, that for some reason these cells, which are also epithelial cells like keratocytes, just from different organisms, they organize this huge contractile actomyosin ring at the very periphery of the cell. So myosin is not bunching up around the cell body. It's remaining at the periphery for completely unknown reasons. My guess is that the adhesions here at the periphery are so strong that they somehow retain this actomyosin at the very periphery. So besides this actomyosin ring, like this huge actomyosin bundle, there are this tiny nascent local lamellipodia which constantly try to emerge at the periphery and die. Okay. So that's very important. So those lamellipodia are trying to protrude and immediately pulled back by this actomyosin ring. So the fascinating thing here is that to polarize the cell, you have to weaken myosin. So remember, in the keratocyte, you have to strengthen the myosin to polarize the cell. Here you have to weaken it. Like in control, the cell never polarizes. But if you weaken myosin, the cell polarizes and starts moving with kind of roughly keratocyte shape.
Quick question: is it cortex? Do you call this actomyosin ring cortex? Or? Okay.
This is probably a question to Eva. It is actomyosin ring, and if it's cortex or not, I don't know.
It's not a sheet, so it's a bit different.
Yeah. I wouldn't call it cortex because in the cortex, to my mind, it's well, cortex is not contractile to my mind. I don't know.
Well, it is differently.
It is different, yeah.
But it's not. It's not. The filaments are not long and aligned, and here it's probably long and aligned.
Probably, yeah. We don't know what the length of
it's a 1D structure versus the cortex being a 2.5D or whatever.
Yeah, yeah. So what explains this polarization mechanism? So another clue is that if you actually inhibit myosin with blebistatin and start looking at the actin in the area of actin-myosin bundle of this nascent lamellipodia, then when myosin is inhibited, the bundle is dissolving, but lamellipodia are becoming stronger. So effectively, there's a competition between actin-myosin bundles and branched lamellipodial actin. So this is the key notion. What I don't have time to describe is many experiments with perturbation of adhesions which seem to have no effect whatsoever on this polarization mechanism, unlike in keratocyte. So adhesions here are, they're probably underlying all of this, but they're not the main players. So the model here, conceptually, is very simple. There's a competition between two actin networks, branched actin and bundled actin. And competition is very simple. If branched actin starts winning, it simply pushes the bundles away from the leading edge which is protruding. So bundles are leaving the leading edge. On the other hand, if bundles start to win, and so they simply compress branched actin into the bundles. So that's so they compete, they kill each other, those two networks. Another thing is that the movement of the boundary affects this competition. If locally the boundary protrudes, then it helps growing branch networks blowing actin bundles away from the leading edge. on the other hand, if the boundary starts retracting, it compresses actin in front of it, creating bundles and disadvantaging branched actin. So that's pretty much it. The essential, and of course, the movement of the boundary is coupled to the densities. If there's a lot of branched actin, the boundary protrudes. If there's a lot of bundles, the boundary retracts. So the key here, again, that there's a feedback between the movement and competition. Plus, there's this competition between two networks. And so when we simulated, so the equations, they look kind of formidable, but in fact, this is the famous, simple competition of two species for common resource model, which is around in mathematical biology, coming from ecology, from Lotka-Volterra, from more than 100 years ago, nothing else. The only thing is it's coupled, it's spatial now, and it's coupled to the movement. That's the key. And so there's actually very beautiful actual mathematics here, which I don't have time to describe. I can talk to mathematicians later about it. But so then simulations show that depending on the parameters, you can get either you can get polarized cell. So the two types of networks segregate. This is the same simulation with noise. So the model makes quite a number of very non-trivial predictions, which all check out. I'll just show you two of them. So one of the predictions is this. We got the phase diagram for this model. How the shape of the cell and the state of it depends on the strength of contraction. So if you increase contraction, you kind of move this way in this phase diagram. And so what the model predicts, which is very non-trivial, it's hard to guess. It's hard to guess without a model, is that for a certain region of high contraction, the cell remains not polarized. Then when contraction is relatively weak, the cell becomes like that. So unlike keratocyte, it's long in the direction of movement. But then you decrease the strength of contraction, the cell becomes more like keratocyte. And when you decrease contraction too much, the cell doesn't polarize again. Okay? And that's the experiment. Increasing concentration of blebistatin, which inhibits myosin. For too weak inhibition, the cell doesn't lose polarity. Then for certain weaker myosin, the cell is long in the direction of movement. Then it's become more and more like keratocyte. And then when myosin completely inhibited, the cell cannot polarize. It's, of course, not remaining round. Basically, what happens, there are protrusions everywhere. They compete and deform the cell into this ugly shapeshifter.
This is actually also the disruption of cytoplasmic coherence in the extreme right, which I'm going to...
Sure, yeah. Sasha will mention that. So there's, as I said, this is the tip of the iceberg of what I'm describing. And this is the experiment which model predicted, and experimentalists confirmed what I'm very proud of, that in a rare occurrence here, we predicted it before the experiment was actually done. And so the prediction is that if you stop the movement, the cell will not be able to polarize with weaker myosin. Okay? Because the movement of the cell is an essential part of these feedbacks. So in this experiment, the cell was put in a shallow well of the size roughly equal to the size of the cell. So the cell cannot move, it bumps into the walls of this well. And so in this case, when blebistatin was applied, when control cell polarized and moved, confined cell, you see that cytoskeleton is not polarized there. So movement is an essential part of this whole process. So that's pretty much it. Here are the conclusion from all the three stories. So sorry, the two last stories were so fast. These are the people who were involved in research. So all the modeling was done by my former postdoc, Kun-Chun Lee. The experiments on keratocytes were done in Julie Thiriot lab in Stanford. The electric field experiments are done in my joint lab with Min Zhao, and Hui Sun was the guy who was making it. Free boundary numerical stuff we did with the help of UCHC team. And the last project was done together with Danuser Lab. So Alexis Lomakin is the guy who did most of the experiments. So this will hopefully come out soon in Nature Cell Biology. Thank you.
And we have time for a few more brief questions, but if there are long things, let's defer them to the discussion 11:30.
It is actually not a question, but a comment. I want to introduce another entity, namely microtubules, because it is well established that in some cell types, like fibroblasts, microtubules are critically important for polymerization. On the other hand, in cells like keratocytes, microtubules are more or less not important. You can disrupt microtubules in keratocytes, they will polarize and will move exactly like with microtubules, but not the same in fibroblasts. I am not sure about these ER2 cells because it might be that they are sensitive.
So in ER2, the short answer is reviewers asked us about it, and we did the classical experiment with nocodazole, no change.
Okay, so in epithelial cells different, but in fibroblasts, obviously, the polymerization depends on microtubules, and until now it is not absolutely clear what is going on. Obviously, it is an interaction between microtubules and actin cytoskeleton in general. One idea is that microtubules affect turnover of focal adhesions. Another idea that microtubule can locally affect contractility, which could be two connected effects, but obviously it deserves also some attention.
Absolutely. So and Sasha, actually, the author of Prager-Kutorsky paper in Nature Cell Biology a few years ago, with yet another mechanism of cell polarization, again suggesting that there's no the mechanism, there are many different mechanisms. And so every new Nature paper tells you that this is the mechanism, but it's not true. This is one more mechanism.
Yeah, I just also wanted to add a little comment about your last story.
You speak up, please.
Yeah, which is that, in fact, I don't know. I mean, of course, if it's the case here, but in your initial model that you showed, if you look at the bifurcation structure, in fact, the phase transition from a static to a motile state is reentrant. Meaning that it's perfectly possible that by applying blebistatin, you switch from a static state to a motile state again. So it's in fact inside the first model that you showed. So if you look at in the phase space of adhesion and contraction, it's like the motile zone is a finger essentially. So in fact, by inducing contractility or applying blebistatin, so caliculin or blebistatin can lead to motion. Depends from where you start. So it's also plausible. I don't know, of course.
Yeah, that's a great comment, and that we didn't think much about it. So Pierre is the expert on those bifurcation for contractile problems. All right, thank you.
Okay, thank you very much.